Alright, so we just talked about the different kinds of amino acids. Now let's go through how these amino acids are broken down. As we've seen, not all amino acids are created equal. Therefore, the different amino acid groups go through different catabolic processes, many of which yield metabolic fuel in the form of pyruvate or acetyl-CoA and ultimately glucose and ketone bodies. But these pathways are not very high yield for your exam. What I do want you to know is that the breakdown of amino acids are going to yield ammonia. And if this ammonia isn't disposed of properly, it can cause problems. So how does our body get rid of the ammonia? Our solution is going to be urea. Urea is a non-toxic, disposable form of ammonia, which is synthesized in the liver and excreted via the kidneys into the urine. Urea is going to account for up to 90% of all nitrogen excreted in the urine. So let's go ahead and take now a look at the urea cycle. The reactants for the urea cycle are going to be ammonia, which I just said, ATP, and also carbon dioxide. So here it's in the form of bicarbonate. The products of the urea cycle are going to be ADP, since we're just consuming that ATP, urea, and fumarate. So we have input and an output of this cycle. It's important to note that the urea cycle takes place in both the mitochondria and the cytoplasm of a cell. The rate limiting step for the urea cycle is going to be the very first reaction, and that's the step that's catalyzed by the enzyme carbamoyl phosphate synthetase 1. This enzyme is going to convert carbon dioxide, so our bicarb, and ammonia into a molecule of carbamoyl phosphate, which can then enter the urea circle and combine with ornithine to create citrulline. The enzyme carbamoyl phosphate synthetase 1 may sound familiar since you heard it before at the beginning of the biochemistry chapter. Just remember that carbamoyl phosphate synthetase 1 is going to be used in the urea cycle, while carbamoyl phosphate synthetase 2 is going to be in pyrimidine synthesis. So you've seen this before. Carbamoyl phosphate synthetase 2 is used in pyrimidine synthesis. Now, once we've created a molecule of carbamoyl phosphate, we are ready to take a spin in the urea cycle. This carbamoyl phosphate is going to combine with an ornithine to create a molecule of citrulline. We sprinkle in an aspartate and an ATP to create an arginosuccinate, and then also we'll get rid of that ATP and create an ADP. Before we keep cycling through, let's quickly talk about the structure of urea. Urea itself has two nitrogenous groups. One of those nitrogenous groups is going to come from the ammonia that we started with, and the other nitrogenous group is going to come from the aspartate that we just added. There's going to be a carbon group in urea, and that's going to originate from the CO2 that we originally added. So eventually, as we keep spinning through, we're going to get our first product out, a fumarate molecule. Where have we seen fumarate before? Well, hopefully you're thinking about the TCA cycle. So let's go ahead and clear some of that off. Here's our TCA cycle. So where have we seen our fumarate before? It's going to be right here. So notice how fumarate can feed back into the TCA cycle, so it's actually not going to get wasted. So let's go ahead and bring back our urea cycle here. So back to the cycle, we notice how our arginine gets turned into an ornithine eventually, which is then used to start our urea cycle off again. Additionally, we get a molecule of urea out, which will travel to the kidney, like I said, for excretion. It's a non-toxic form, it'll get excreted by the kidney. We have now successfully gotten rid of an ammonia group. And if you're following along in the First Aid Step 1 book, take a look at the urea cycle figure. It's important to note that this process requires two ATP molecules. I'll be discussing the urea cycle disorders in just a second, so bear with me. Let's just take this one step at a time and first test our knowledge of what we just learned about the urea cycle. The urea cycle ultimately yields blank, which is then excreted in the urine, and blank, which can enter the citric acid cycle, or the TCA cycle. Now that you're experts on the urea cycle, you know that the urea cycle is meant to excrete urea into the urine, which is getting rid of the toxic ammonia form from amino acid breakdown. The urea cycle also creates a molecule of fumarate, which can then feed into the TCA cycle. So this will go into the urine, and this will go into TCA, Krebs, or citric acid cycle. Now that we've figured out how to get rid of ammonia, let's now figure out how we get that ammonia produced from the body to the liver cytoplasm where the urea cycle is going to take place. As I mentioned before, ammonia is a toxic molecule that must be converted to urea so that it can be excreted in the urine. However, the urea cycle is only going to occur in the liver, which means that the ammonia that gets created by the periphery must first be transported to the liver. So how does this toxic ammonia get from our body to our liver? Well, the solution is going to be in the form of alanine, which is shown here. In the muscle, so we have our little pretty muscle here, the amine group that you're trying to get rid of is first added to an alpha-ketoglutarate in a transamination reaction, which is going to result in the formation of a glutamate. By the way, this is the same alpha-ketoglutarate that we talked about in the TCA cycle. In a second transamination reaction, this amine group is transferred to pyruvate, which is going to form our molecule of alanine seen here. This is catalyzed by the enzyme alanine aminotransferase, or ALT for short. This is the same enzyme that you measure for hepatic function testing. Alanine can then circulate through the blood via the Cahill cycle. So the alanine is going to transfer now in the bloodstream in the form of alanine and make its way to the liver. Once in the liver, the previous reaction is going to be reversed. So here we made our alanine and now we're going to unload our alanine. And the amine group is transferred from that alanine to an alpha-ketoglutarate to form a glutamate molecule. In the final step, the ammonia from our glutamate is going to enter the urea cycle, and thus we're going to spin just like we saw in the last slide, complete the urea cycle. This allows glutamate to cycle back to form our alpha-ketoglutarate. So what happens if you have too much ammonia? In other words, you're producing this ammonia faster than you can get rid of it. Excess ammonia in the blood, called hyperammonemia, is a serious medical condition that causes hepatic encephalopathy. These patients will present with symptoms such as asterixis, which is a tremor, slurring of the speech, somnolence, vomiting, and blurry vision, and it can also be deadly due to the cerebral edema. It can be caused by a few different problems. Hyperammonemia can be caused by liver disease, since that's where the ammonia is broken down, or it can be due to a genetic deficiency of a urea cycle enzyme, which is going to prevent the urea cycle from actually working properly. In either case, ammonia is going to build up and it'll be transferred to alpha-ketoglutarate to form a glutamate, as we talked about earlier. This is going to deplete your supply of alpha-ketoglutarate, since you're going to be combining the ammonia now with alpha-ketoglutarate to form glutamates. When this alpha-ketoglutarate supply is depleted, which metabolic cycle is going to be inhibited? Well, hopefully you're thinking you saw alpha-ketoglutarate in the TCA cycle. So the TCA cycle will be inhibited when you use up all your alpha-ketoglutarate. And then lastly, how are we going to treat this hyperammonemia? The treatment of this condition is to limit the protein in the diet. If you want to look like a rock star on clinical rotations, you should also be aware that you can give lactulose to acidify the GI tract. When you give lactulose, the ammonia is going to get converted into ammonium, NH4+, and this ammonium cannot cross the gut epithelium. The nitrogen is now trapped in the GI lumen and it'll now be excreted rather than absorbed. Other options include giving rixamin, which is going to decrease the bacterial load in the gut that produces ammonia. Amino acid binders such as benzoate or phenylbutyrate can also increase the excretion of the ammonia. Let's now take a quick look back at the urea cycle. We've already seen this figure before. Do you remember what the rate limiting step of the urea cycle was? The rate limiting step is the reaction formed by carbamoyl phosphate synthetase 1, which is going to take an ammonia group and a CO2 group and turn it into a carbamoyl phosphate. Now, how does the body regulate this enzymatic step? N-acetylglutamate is going to be the answer. N-acetylglutamate is a cofactor for the enzyme carbamoyl phosphate synthetase 1. So this is going to act as a cofactor for this enzyme. Now, if N-acetylglutamate cannot be synthesized because you're missing this N-acetylglutamate synthase enzyme, just sound it out, it's going to synthesize N-acetylglutamate, then the rate limiting step for the urea cycle is inhibited and hyperammonemia can occur. You're going to get a buildup of these reactants. So your ammonia levels will increase in the body. You're not able to get rid of it. So an N-acetylglutamate synthase deficiency is going to present the same as a carbamoyl phosphate 1 synthase deficiency. However, if you have normal urea cycle enzymes with an increase in ornithine, so if you have an increase in ornithine levels, then this can suggest that an N-acetylglutamate synthase deficiency is present. In both N-acetylglutamate synthase deficiency and carbamoyl phosphate synthase deficiency, you would see symptoms early in life, such as irregular breathing, poor temperature regulation, feeding difficulties, developmental delay, or intellectual disability. Speaking of urea cycle enzyme deficiencies, ornithine transcarbamylase deficiency is going to be the most common urea cycle disorder. A lot of the enzyme deficiencies that I've mentioned before are autosomal recessive. However, this one's actually going to be X-linked recessive. So I've got a question for you. Can a male with ornithine transcarbamylase deficiency, which is an X-linked recessive disorder, can that male pass this trait off to his children? Well, let's think about this. Well, yes, he can pass the faulty X chromosome to his daughters, so yes to the daughters, but he cannot pass it off to his sons. Since it's an X-linked recessive disorder, his daughter is also going to have to receive a faulty X chromosome from the mother as well to actually have the disease of a true ornithine transcarbamylase deficiency. However, a male is not going to pass off an X chromosome to his son, so he's not going to pass it off to the son, but daughter, he can pass off the trait. Remember, the enzyme ornithine transcarbamylase is normally used to convert an ornithine and a carbamoyl phosphate to make a citrulline. So, if you don't have this enzyme, then carbamoyl phosphate levels can build up. So manifestations of ornithine transcarbamylase deficiency include orotic acid in the blood and urine, a decrease in the blood urea nitrogen, since remember, urea cannot be formed without this enzyme to be present. So no urea would be formed. And then also you'd get the symptoms of hyperammonemia because you're going to have ammonia that cannot be converted into urea. There's also another disease besides ornithine transcarbamylase where you're going to see orotic acid in the urine. Do you remember what that is? Well, that disease would be orotic aciduria, which is caused by a defect in one of the enzymes that converts orotic acid into UMP. But do you remember, how can you differentiate between orotic aciduria and an ornithine transcarbamylase deficiency? In the disease orotic aciduria, you're not going to have hyperammonemia, since ammonia is excreted via the urea cycle just fine. In ornithine transcarbamylase deficiency, since you can't excrete the nitrogenous waste via the urea cycle, you're actually going to get ammonia that builds up in the blood. So this is going to be your difference between orotic aciduria versus an ornithine transcarbamylase deficiency. In orotic aciduria, you will not have hyperammonemia. While in ornithine transcarbamylase deficiency, remember I said that ammonia is going to get built up in the system because you're not able to get rid of it, so you're going to have increased ammonia levels. Let's try our hand at a flash quiz. A lethargic newborn boy with a low BUN is diagnosed with the most common urea cycle disorder. What substance will you ultimately find in this baby's blood? This one's a tricky one. Hopefully you're thinking orotic acid, which is used for pyrimidine synthesis. Let's talk this one through. The child has a ornithine transcarbamylase deficiency. So in this figure here, we have our ammonia, which gets converted into a carbamoyl phosphate. Now this carbamoyl phosphate has two options. It can go downwards in this figure to go into the urea. Let's see, urea cycle. So here it'll get combined with ornithine via the enzyme ornithine transcarbamylase to form a citrulline and then ultimately urea. However, if that enzyme, ornithine transcarbamylase, is deficient, which our newborn has, this carbamoyl phosphate can then enter a different pathway. It can be used to build pyrimidine nucleotides. So let's go ahead and take this one a little bit further. Carbamoyl phosphate can also be used to build pyrimidine nucleotides, which I just said. So what are those three pyrimidine nucleotides? Well, you learned these earlier in the biochemistry molecular section. These will include your cytosine, thymine, and uracil. So here we can see two pathways side by side. These are going to be both metabolic processes that use carbamoyl phosphate, and we're going to look at them together. So here we see our carbamoyl phosphate can enter the urea cycle. However, if our ornithine transcarbamylase enzyme is not there or it's defective, our carbamoyl phosphate will then enter the pyrimidine synthesis pathway. So if we have a urea cycle deficiency, such as an ornithine transcarbamylase deficiency, this carbamoyl phosphate will switch pathways, leading to an orotic acid buildup. You'll also see a high orotic acid buildup in the urine in orotic aciduria, which is caused by a defect in one of the enzymes that converts orotic acid into UMP. But do you remember, how can you differentiate between an ornithine transcarbamylase deficiency and orotic aciduria? Well, orotic aciduria will not have hyperammonemia since you can still break down the ammonia just fine.